Imagine sitting in a clinical sterile doctor's office and, well, you are handed a diagnosis that just stops your heart.
Right.
A highly aggressive, life-threatening blood cancer. By any metric, it is basically the worst possible news. But what if that specific cancer diagnosis and, the grueling, almost lethal treatment required to fight it ended up being the exact key to curing you of an entirely different, previously incurable viral disease you had been managing for a decade?
I mean, it sounds completely paradoxical. As a society, we tend to categorize diseases as strictly destructive forces, just isolated villains.
Right.
The idea that introducing a catastrophic cellular breakdown could somehow neutralize a deeply entrenched viral infection, well, it challenges our fundamental understanding of how the body handles trauma.
But that is exactly the reality of what happened to the Berlin patient. So welcome to this deep dive. Today, we are exploring the raw, unhyped biology of how our DNA interacts with the viruses around us. We're going to unpack the mechanics of human immunity, look at how viruses function essentially as microscopic genetic poison, and dissect the incredible moments where our own genetic mutations can turn a virus's ultimate weapon against itself.
And we are approaching this strictly through the lens of established genetic science. So, no sensationalism, no marketing language. The reality of cellular biology, the microscopic war happening inside you every single day, it doesn't need exaggeration to be fascinating.
It really doesn't. So let's get right into the human element here, because Timothy Ray Brown's medical reality anchors all the complex genetics we're about to discuss.
Right.
We are talking about an American who was studying in Berlin when he was diagnosed with HIV. And thanks to the antiretroviral therapies available a couple of decades ago, he was actually living a fairly functional life. For 10 years, the medication kept the virus suppressed.
But suppression is not a cure. The virus is always there, kind of waiting in the background.
Yeah.
And then, after experiencing a prolonged period of intense physical weakness, Timothy receives a secondary diagnosis, which was leukemia.
Yeah, and let's not gloss over what leukemia actually does, because it is crucial for understanding the extreme measures his doctors had to take. The bone marrow is essentially the biological factory for your white blood cells.
Right. It's supposed to be an orderly assembly line.
But in leukemia, that assembly line just goes completely rogue.
Precisely. Those white blood cells begin dividing uncontrollably. And because the space inside your bone marrow is finite, these rapidly multiplying defective cells physically crowd out the normal, healthy blood cells.
Wow.
They consume all the resources. So the patient experiences severe anemia, overwhelming weakness, and ultimately, the total destruction of the immune system. The body is essentially suffocating on its own defective cellular production.
Which means you can't just treat the symptoms, you have to go to the source. You literally have to shut down the factory. And the method for doing that is chemotherapy. Now, I think a lot of people view chemo as a targeted strike, but in this context, it's more like a scorched earth campaign. You are using highly toxic substances to systemically destroy all the fast-growing cells in the bone marrow.
And the collateral damage is immense. The treatment doesn't easily distinguish between a fast-growing cancer cell and a fast-growing healthy cell. So, hair follicles, the lining of the digestive tract, and the remaining immune system are all completely devastated.
It's basically the biological equivalent of burning down an entire forest just to eradicate a highly invasive, deep-rooted weed, and then crossing your fingers that the soil isn't too toxic to plant new trees afterward.
That analogy captures the brutality of the necessity perfectly. You have to bring it all down because if even one microscopic malignant cell remains, it divides.
Right, the math of it.
Yeah, 1 becomes 2, 2 becomes 4, 8, 16. The exponential math of cancer means that a single surviving cell can repopulate the entire disease. In fact, the sheer toxicity of this treatment carries its own massive risk. About 2.5% of cancer patients die from the chemotherapy treatment itself, purely due to drug intolerance. Their bodies simply cannot survive the poison.
That is sobering. And Timothy Ray Brown had to endure this scorched earth campaign twice. His cancer actually returned after the first round.
Yeah.
So they had to subject his body to this incredibly toxic treatment a second time, completely wiping out his bone marrow.
And once you have successfully eradicated the bone marrow, you are left with a patient who has absolutely no immune system. A common cold would be lethal at that point.
Right.
This brings us to the replanting phase of your forest fire analogy, which is the stem cell transplant. You take healthy stem cells from a genetically compatible donor's bone marrow, introduce them into the patient's bloodstream, and wait. Those donor cells migrate to the bones, multiply, and begin the arduous process of building a brand new immune system from scratch.
Okay, so we have the baseline. Timothy's immune system is wiped clean, and he is about to get a transplant. But to understand why his new immune system ended up curing his HIV, we have to look at the enemy, right? We need to look at what HIV actually does when it enters the body.
It helps to trace its evolutionary origins first. The human immunodeficiency virus was first definitively diagnosed in 1959 in what is now the Democratic Republic of Congo.
Okay.
But it didn't just appear out of nowhere. It evolved from a very similar pathogen found in chimpanzees known as simian immunodeficiency virus, or SIV.
And the mechanism of how it got to chimps in the first place is just wild. I mean, researchers traced it back to chimpanzees hunting and eating two different smaller ape species.
Yes. The leading scientific consensus is that a chimpanzee consumed two different species of monkey, each carrying a distinct virus. Inside that one unlucky chimpanzee, those two viruses encountered each other, merged their genetic material, and mutated into a new, highly effective supervirus.
Wow.
Decades later, around 1920, that virus made the jump to humans.
And it became one of the most devastating pandemics in modern history. At its peak in 1995, there were 3.5 million new infections globally every single year.
Yeah.
It's down to about 1.5 million now, which is obviously still massive. But what I want to zero in on is the physical scale of this threat, because the disparity between the size of the virus and the damage it causes is hard to wrap your head around.
The scale is almost incomprehensible. Let's visualize it. If we imagine a single human white blood cell scaled up to the size of a skyscraper, where the nucleus alone is 6 meters across, the HIV virus approaching that skyscraper would only be the size of a tangerine.
A tangerine taking down a skyscraper. That's crazy.
Exactly. And it is stunningly simple in its construction. The human genome contains billions of base pairs. HIV consists of just 9,749 genetic letters.
Just under 10,000 letters.
Right. Those letters make up only 9 genes. So if human DNA is a massive multi-volume library of complex instructions, the entire genetic instruction manual for HIV fits on just 10 pages.
10 pages of code. I mean, that pushes right up against the philosophical question of whether a virus is even a living organism. It doesn't eat, it doesn't metabolize. It's essentially just a malicious 10-page script sent to a cellular factory.
It is purely information, wrapped in a delivery vehicle. And its interaction with our cells is a precise 3-step hijacking. Step 1 is incorporation. The virus breaches the human cell and literally injects those 10 pages of code into the cell's nucleus, forcing the human genome to integrate the viral genes.
It edits our own source code.
Yes. Step 2 is the build phase. The human cell, now compromised, blindly reads these new instructions. Instead of doing its normal job, the cellular machinery is repurposed to manufacture thousands of empty viral envelopes, which look like tiny microscopic spheres.
Okay.
Finally, step 3 is copy and fill. The cell frantically copies the viral DNA, stuffs those copies into all the newly built spheres, and continues doing this until the physical volume is too much. The human cell literally bursts open, dying in the process, and releasing thousands of new viruses to go infect the neighboring cells.
It is a microscopic heist. It hacks the factory, uses all the resources to build its own army, and then blows up the building on the way out.
Perfectly stated.
But, and here's the crux of the entire Berlin patient story. To execute that heist, the tangerine has to get inside the skyscraper first. It can't just float through the walls. It requires a very specific door.
In molecular terms, that door is a receptor on the surface of white blood cells known as CCR5. HIV is highly specialized. It actively seeks out this specific CCR5 receptor, binds to it, and uses it as the entry point to inject its genetic payload.
Okay, this brings us back to Berlin, to a doctor named Jörg Hutter at the Charity Hospital. Dr. Hutter is treating Timothy Ray Brown. He knows Timothy's bone marrow has been destroyed by the chemo, and he needs a stem cell transplant. Now, typically, doctors look at a donor registry and just try to find a basic genetic match to avoid tissue rejection. But Dr. Hutter takes a completely different angle.
Right, he looked at the UK registry, which contained hundreds of thousands of potential donors. For Timothy, there were 267 compatible matches. But Dr. Hutter recalled a specific anomaly in human genetics. He knew that a small fraction of the population is naturally resistant to HIV. And they are resistant because they carry a genetic defect in the specific gene responsible for building that CCR5 receptor.
So Dr. Hutter thinks, what if I don't just give Timothy a new immune system? What if I give him an immune system built from the DNA of someone whose cellular doors are already broken? He takes those 267 compatible donors and actively screens their genetics for this specific defect.
It was an incredible long shot. But he found one, donor number 66. This donor didn't just have one defective copy of the gene, they had a defect in both copies. The one inherited from their mother and the one inherited from their father.
The double defect, which means the body doesn't even have a backup plan.
Correct. Because of this double mutation, every single white blood cell produced by donor 66 featured severely malformed CCR5 receptors.
So the door was essentially missing the handle and the hinges.
Exactly. To the HIV virus, the cell was impenetrable and had nothing to bind to. So on February 7, 2007, the medical team transplanted donor 66's stem cells into Timothy Ray Brown. As those stem cells engrafted and began producing a brand new immune system, every single white blood cell that populated Timothy's body had this broken door.
So the virus is still floating around in his system looking for a way in, but the locks have all been changed.
And without a host cell to hijack, a virus cannot replicate. It just degrades. 3 months after the transplant, they tested Timothy's blood. The HIV was completely undetectable. By replacing his immune system with mutant-resistant cells, he became the first person in the world to be cured of HIV.
The mechanics of that are undeniably brilliant, but as someone looking at this from a broader perspective, I have a massive structural question.
Sure.
If this double gene defect is a literal force field against one of the deadliest viruses on the planet, why doesn't everyone have it? How did donor 66 get so lucky?
That is an evolutionary puzzle that scientists had to untangle. The statistics of this mutation are fascinating because they are fiercely localized. This specific CCR5 defect is uniquely European. It is practically non-existent in African and Asian populations.
Really?
Yes. In the UK, for example, 1 in 5 people carries one defective copy of the gene, which gives them a lowered risk of infection. And 1 in 70 people has the double defect, granting them full resistance to the main R5 variant of HIV.
Wait, 1 in 70. That's a couple of people in every crowded subway car in London walking around basically bulletproof to this virus.
It is.
And the genetics trace this mutation back to Scandinavia roughly 2,000 years ago. But evolution is driven by survival. A mutation only spreads that widely if it provides a massive survival advantage against a deadly threat, right?
And we know that threat wasn't HIV. HIV has only been infecting humans for about a century. 2,000 years ago, HIV didn't exist in the human population, so the virus couldn't have shaped our genetics across a whole continent.
So if it wasn't HIV driving this mutation, the obvious next guess is the Black Death. I mean, the bubonic plague wiped out huge swaths of Europe around 1350. It killed millions. That's exactly the kind of evolutionary bottleneck that alters human DNA permanently.
It's a logical hypothesis, but the biology doesn't actually align. First, the Black Death is caused by a bacterium, Yersinia pestis, not a virus. It doesn't utilize the CCR5 receptor to infiltrate cells at all. Furthermore, from a genetic timeline perspective, 1350 is just too recent. A mutation couldn't achieve a 1 in 5 saturation rate across Europe in just a few hundred years.
Okay, so cross off HIV. Cross off the plague. What ancient pathogen was slaughtering Europeans but sparing the few people who randomly had a broken CCR5 receptor?
The scientific consensus points to a pathogen we usually associate with childhood, which is chickenpox.
Wait, chickenpox? Like the itchy rash?
The varicella-zoster virus, yes. Historically, before modern medicine, this virus hit the Scandinavian region incredibly hard, and it was highly lethal. And here is the crucial biological overlap. Just like HIV, the chickenpox virus targets white blood cells, and it uses that exact same CCR5 receptor to gain entry.
That is wild. So 2,000 years ago in Scandinavia, there's a brutal outbreak of chickenpox. People are dying in large numbers, but a small handful of people have this random genetic glitch, a broken cellular door, so they don't get severe chickenpox. They survive.
They survive, they reproduce, and they pass that broken door gene down to their children. Over generations, aided by what geneticists call the founder effect during periods of colonization and migration, this specific mutation spread widely among people of European descent.
Let's unpack the founder effect briefly for you listening. It basically means that if a small group of people migrates to a new area, and just 1 or 2 of those founders happen to have this rare gene, well, as their population expands in isolation, that rare gene suddenly becomes highly common in the new population.
Exactly. It is a remarkable evolutionary accident. A brutal historical battle with chickenpox essentially forged a biological shield. And modern humans inherited that shield entirely by chance, protecting them against an entirely different retrovirus that wouldn't even exist for another 2 millennia.
It really is an evolutionary lottery. But that brings me back to the present day. We have the Berlin patient. The biology is proven. We know how to lock the door. So why is this not the standard of care? Why are millions of people still living with HIV if we have the blueprint for a cure?
Because the reality of medical intervention is often constrained by immense risk and logistical impossibility.
Yeah.
We have to confront the 5 major roadblocks to this treatment.
Okay.
First, the conditions for this procedure are incredibly strict. You cannot just ethically wipe out someone's healthy bone marrow. Dr. Hutter actually treated 6 other patients who had both HIV and cancer using this exact method. Tragically, all 6 died from either the underlying cancer or severe transplant complications.
That's awful.
Yeah, the procedure is only medically justifiable if the patient already has lethal cancer and desperately requires bone marrow eradication anyway.
Which immediately leads to the second roadblock, extreme mortality risk. We talked about the 2.5% death rate just from the chemotherapy. But when you factor in the stem cell transplant itself, the body potentially rejecting the new cells, or the complete lack of an immune system during the transition, the fatality rate for the transplant is a staggering 25%.
So before you even know if the HIV cure worked, the patient faces a 12 to 30% risk of dying strictly from the procedure. It is a massive life-threatening gamble.
I have to push back a bit on the framing of this as a cure for the general public then.
Yeah.
If you have up to a 30% chance of dying on the operating table, that is statistically much more dangerous than just living with the virus using modern medicine.
You are entirely correct. And even if a patient accepts that massive risk, they run into the third roadblock: donor availability. Finding a stem cell match is difficult enough. If you are of European descent, you have about a 77% chance of finding a basic match. But remember, you don't just need a basic match. You need a match who also possesses that rare 1 in 100 double genetic mutation.
It's finding a needle in a stack of needles.
And that's only for Caucasians. For other ethnicities, the math is far worse. Hispanics and Asians only have a 50% chance of finding a basic genetic match, and people of African descent have just a 20% chance. And again, the CCR5 mutation is virtually non-existent in those populations anyway.
And if you magically find a donor and survive the risk, you hit roadblock 4, which is the staggering financial cost. We are talking about $200,000 in the UK and upwards of $700,000 in the US. It is prohibitively expensive.
Very much so.
Which brings us to the fifth and perhaps most important roadblock. The alternative therapies today are just incredibly effective.
They are. A 20-year-old infected with HIV today who adheres to modern combination therapies has an average life expectancy of 75 years. That is barely a difference compared to the 80-year life expectancy for someone without HIV.
Wow, 75 years.
Yes, the modern medications have minimal side effects, they keep the viral load undetectable, and they drastically minimize the chance of transmission.
For almost everyone, the daily pill is infinitely safer than the bone marrow firebomb. And I think we need to ground this in the ultimate reality of Timothy Ray Brown's life. He was cured of HIV, yes, but the underlying cancer story didn't end there.
No, it didn't. On September 29, 2020, after a grueling 5-month battle, his leukemia returned and he passed away. His HIV remained cured until the very end, but the incredibly aggressive cancer that necessitated the treatment in the first place ultimately claimed his life.
It is a sobering reminder of how fragile human biology really is. But Timothy's legacy forced a paradigm shift in genetics. We just spent 20 minutes discussing viruses as lethal hijackers, as microscopic poison. But scientists looked at this mechanism and asked a brilliant question. If a virus is nature's absolute best mechanism for breaking into a human cell and seamlessly inserting new genetic code into our DNA, can we harness it?
That question is the entire foundation of gene therapy. If the virus already possesses the molecular machinery to bypass our cellular defenses, why not weaponize it for our own benefit? If we can remove the malicious, disease-causing code from the virus, could we replace it with helpful corrective code?
But hold on, if we are calling a virus nature's perfect delivery system, isn't that ignoring the fact that our immune systems are designed specifically to destroy them on sight? You can't just inject a virus and expect the body to welcome it.
That is the core challenge. But the theory was first successfully executed in 1990, moving from concept to reality, with a 4-year-old girl named Ashanti da Silva. She suffered from a severe genetic disorder called ADSCID. Essentially, she was born entirely without a functioning immune system.
Oh, wow.
It's a condition commonly referred to in popular culture as bubble boy disease. Because her body couldn't produce the ADA enzyme, toxic byproducts built up and destroyed her immune cells. Every common bacteria or minor virus was a lethal existential threat.
So doctors devised a radical plan. They took a virus and they essentially hollowed it out. They completely removed the viral instructions, the build more viruses and explode command, and in its place they inserted a healthy, functioning human gene, the exact AD gene that Ashanti was missing.
Then, they extracted some of Ashanti's own defective white blood cells, and in a controlled laboratory environment outside her body, they let this genetically modified virus infect those cells.
And the virus did what millions of years of evolution trained it to do. It broke into the cell, navigated to the nucleus, and inserted its payload into her DNA. But this time, it wasn't dropping a bomb. It was delivering the missing blueprint for a healthy immune system. They infused those newly programmed cells back into her body, and it actually worked. Her body started producing the missing enzyme.
It was a monumental triumph for early genetic science. She required regular top-up injections, but she could live a relatively normal life. However, as is often the case when we manipulate systems we barely understand, there were unforeseen catastrophic consequences in subsequent trials.
This is the part of the science that is just heartbreaking. 10 years later, doctors conducted a larger trial. They used a similar viral technique on 20 children suffering from a closely related immune deficiency called IL-2RG-SCID. Initially, it looked like a total victory. All 20 children responded perfectly. The engineered virus delivered the healthy gene, and their immune systems came online. But a few years later, 5 of those children developed leukemia.
They cured a fatal immune deficiency, but inadvertently triggered a highly aggressive blood cancer.
Why does that happen? I mean, if the virus only contains healthy human code, how does the delivery process itself cause cancer?
It comes down to the brute force mechanics of viral insertion. Human DNA is incredibly long and densely packed. When the virus inserts the new, healthy gene into the host's genome, it doesn't just neatly slide it in. The virus literally has to sever the existing DNA strand and forcefully squeeze the new code into the gap.
It's like trying to fix a typo in a massive computer program by randomly pasting a line of code into a file. You might fix the typo, but if you accidentally paste that line right into the middle of the operating system's core safety protocol, the whole computer crashes.
That is exactly what happened. The virus is highly efficient at inserting genes, but in those early trials, it was completely indiscriminate about where it placed them. In those 5 children, the virus randomly inserted the new, healthy immune gene directly into the middle of existing crucial anti-cancer genes.
It shattered the body's natural defense against tumors.
Yes. By breaking those protective genes to make room for the new code, the cells lost their ability to regulate division, turning them cancerous. Medical teams had to intervene. 4 of the children were eventually cured of the leukemia using chemotherapy, but tragically, 1 child died.
So the obvious question for geneticists today is precision, right? Can we tell the virus exactly where to place the new code so it doesn't break a crucial tumor-suppressing gene?
The scientific community is intensely focused on that exact hurdle. The material we are analyzing today clearly outlines this precision problem as the defining challenge of modern gene therapy. Scientists are actively developing methods to pinpoint the exact insertion location to prevent these catastrophic side effects. The path forward absolutely requires exact targeting, avoiding the random splicing that caused the leukemia.
It is the ultimate microscopic surgery, and the stakes couldn't be higher. So bringing this all back to you, the listener, what does this deep dive actually mean for your own biology? Well, the statistics are striking. If you are of Caucasian descent, there is a 19.4% chance you're walking around with one defective CCR5 gene right now.
Right.
And there's a 1.39% chance that you possess the double defect, making your cells physically impenetrable to the main R5 variant of HIV.
However, we must be explicitly clear regarding medical behavior. Genetic resistance is not a shield against all pathogens, nor is it a license for risk. The X4 variant of HIV still exists and utilizes a completely different cellular receptor to enter the cell, not to mention the myriad of other diseases that ignore the CCR5 receptor entirely. Safe habits remain universally essential.
Absolutely. But if you do happen to carry this specific mutation, joining a bone marrow donor database is highly recommended. Because of the sheer rarity of the double mutation, your specific biology could literally be the 1 in a million miracle for someone else facing the dual nightmare of cancer and HIV, exactly like donor 66 was for the Berlin patient.
It is an incredibly profound way to contribute to the medical legacy that began with Timothy Ray Brown.
It really is. And it leaves us with something pretty wild to think about as you go about your day. The line between a deadly pathogen that destroys societies and a highly advanced, life-saving genetic tool is incredibly thin. Literally just a few lines of molecular code separating the two. It forces you to wonder, considering the brutal evolutionary history of humanity, what other ancient, random genetic defects might be hiding in your DNA right now, quietly waiting to protect you from a disease the world hasn't even encountered yet.